Welcome everyone to this ACARE LevelUp session number 7. Today we've got a truly fascinating program for you, delivered by 3 renowned international speakers. I'm going to introduce them now so that we don't interrupt the flow of the program. The first speaker is Professor Marc Riedl from the University of California, San Diego, USA. His discussion will be followed by Professor Danny Cohn from Amsterdam, UMC, University of Amsterdam in the Netherlands. And then we'll have Professor Hilary Longhurst from the University of Auckland and Department of Immunology, Auckland City Hospital, New Zealand. Today, our learning objectives include exploring the fundamentals and advancements of gene editing with a special focus on CRISPR-based technologies and their potential to revolutionize genetic medicine. We'll also examine the application of CRISPR-based gene editing in treating hereditary angioedema, understanding the clinical progress we're making, the challenges and future potential for improving patient outcomes. This is our agenda. We'll have our first speaker, which is a video of Marc because of the time difference, so there won't be a discussion after his presentation, but with the other 2 talks, we will have a few questions after each one. So we're going to look at the current HAE treatment landscape and discuss how and where gene editing fits in. Danny will discuss CRISPR technology. What is it and how does it work? And then we'll hear from Hilary, who will discuss the results from the clinical trials thus far using CRISPR technology in HAE treatment. So without further ado, we'll move to our first speaker, Marc, who will now go through his presentation by video. Thank you.
Hi, everyone. I'm Dr. Marc Riedl from the University of California in San Diego. It's my pleasure to join this seminar today and give a brief presentation on the current HAE treatment landscape. How and where does gene editing of pre-kallikrein fit? So my colleagues, Dr. Cohn and Dr. Longhurst, are going to present on gene editing technology specifically. My job is really to just give you an overview of the treatment landscape. As we think about new therapies for HAE, it's important to understand sort of where we are and where we're going in terms of clinical development of HAE therapies. So I'll start by just reminding you that we've come a long way in the last several years with HAE treatments. We've moved from non-specific treatments like androgens and antifibrinolytics to very targeted therapies. And as you know, these therapies target the contact system, the kallikrein-bradykinin pathway. And so we have a number of strategies. You can replace C1 inhibitor with current therapies. You can target kallikrein either to prevent attacks or to treat attacks acutely. And you can block the bradykinin B2 receptor, specifically used to treat HAE attacks on demand. And so as I walk through this, we understand, of course, not all of these treatments are available in all parts of the world, but these do represent very important advances that have improved our ability to prevent or treat HAE attacks and certainly improved the lives of our patients and families that have hereditary angioedema. We know from the evidence-based guidelines that these therapies really fit into different treatment strategies. At least that's how they're studied in clinical trials. And a reminder that first and foremost, every patient with HAE needs an on-demand treatment. All patients need to have access to effective on-demand therapies and know how to use those in order to treat these unpredictable, debilitating, and life-threatening attacks. On the right side of this slide is long-term prophylaxis, and we're going to spend a considerable amount of time in this seminar discussing long-term prophylaxis, as this is where a lot of the developmental strategies have really been targeted, given that the understanding that if we can prevent HAE patients from getting sick, from having these attacks, we can really improve their quality of life and control the condition. And finally, short-term prophylaxis, which is utilized before procedures or certain events in life that are known to be high risk for triggering HAE symptoms. This is a table of the studied HAE acute therapies. I'm not going to go into this in detail, but it's important to recognize we have a number of proven treatment options, including C1 inhibitor concentrates, either plasma-derived or recombinant C1 inhibitor. These are given intravenously at the time of an HAE attack and have been shown to effectively stop those attacks rapidly. We also have subcutaneous drugs, ecallantide, which is fairly limited in its distribution around the world, but has been used in the United States to treat HAE attacks as a plasma kallikrein inhibitor. And icatibant, which is used fairly widely around the globe as a bradykinin B2 receptor antagonist, this again being a subcutaneous medication that has been shown to effectively stop HAE symptoms. And so knowing these medicines, their route of administration, their potential side effects, very important in the management of HAE. And again, depending on the region or country that you practice, may have some or unfortunately none of these in some countries. But these are sort of the first and most important step to recognize in managing HAE. Now, long-term prophylactic therapies have changed quite a bit in recent years. And at the bottom of this slide, you see some of the older second-line treatments based on the evidence-based guidelines, androgens or antifibrinolytics. These can be effective and they're still used in some parts of the world due to their efficacy. But of course, we recognize for androgens, there are potential long-term side effects that can be very concerning and serious. And for antifibrinolytics, the data for efficacy, certainly for HAE due to C1 inhibitor deficiency, is not as robust. Increasingly, and based on the evidence that we have, first-line treatments are recommended, and those are the first 4 medications in this table, either plasma-derived C1 inhibitor used intravenously or subcutaneously, lanadelumab as a targeted monoclonal antibody against plasma kallikrein, and berotralstat, a targeted oral kallikrein inhibitor given daily. And again, you see each of these have potential side effects, potential advantages and disadvantages, and we recognize, again, access to these as variable based on the country or region around the world. Just to show you a little bit of data on where we stand right now with modern long-term prophylactic therapies. This is the data from the pivotal trial on sub-Q C1 inhibitor for HAE prophylaxis. Again, this given generally twice weekly. And you can see at the higher dose of 60 units per kilo, this robust efficacy in preventing HAE attacks, a 95% median reduction compared to placebo for this particular treatment approach. Lanadelumab is a subcutaneous monoclonal antibody. This is given once every 2 to 4 weeks. You can see 3 different doses were studied in this phase 3 randomized study that was published in 2018. And at the highest dose of 300 milligrams every 2 weeks, again, this very strong efficacy of over 86% reduction in mean attack rate compared to placebo. Berotralstat is the oral kallikrein inhibitor. It's taken daily, and this is the phase 3 data published in 2020. Again, you can see on across the population, the 150 milligram dose of the medication had a 44% reduction in the mean attack rate compared to placebo. So not quite at the level that we see with the parenteral medications. However, if you look at the responder analysis from this data, you can see in the middle there, a 50% reduction in HAE attack. 50% of the patients had at least a 70% reduction in attack rates. And so this can be a very effective medication for patients, certainly those that prefer oral medication over parenterally administered therapies. Now, if we look on the whole, the goal of treating HAE attacks and certainly of long-term prophylaxis is to improve quality of life. And importantly, we have a number of quality of life instruments that have been used in trials and shown that these modern therapies do in fact improve quality of life. I'm showing this most recent HAE quality of life instrument developed in the United States, not because it's necessarily better in any way. We have a number of excellent tools, but I do want to point out that this quality of life tool incorporated access to medication is a very important domain in determining quality of life for our patients, certainly in the US population. And I think access to medication, whether that has to do with the health system, seeing a specialist, or the chronicity of using these medications can in fact have a huge impact on patients. So adhering to the treatment, having access to the treatment reliably, does affect patient quality of life, at least in this validated instrument, along with the things that we know to be important, the ability to carry out activities, emotional well-being, embarrassment, and so forth. And so I think we also have to understand that there is a burden of treatment for HAE therapies. This was a survey done, again, in a US population, but recognizing that there are treatment-related concerns. You can see that in this particular survey, on total, 76% of the patients tried not to think about the demanding nature of HAE treatment, and 89% of the total patients reported that they had learned to tolerate some difficult aspects of their HAE treatment. So that's not to say there aren't clear benefits, significant benefits from HAE treatment, but we are conscious of the burden of treatment. Again, HAE being a lifelong condition at this point, and the chronic nature of both the symptoms and the therapies that our patients undertake to manage their condition. So with that being said, quality of life and burden of treatment being important, we know that there's been considerable ongoing work, clinical development for other therapies in the HAE space. And this diagram just outlines those strategies. You can see monoclonal antibodies, garadacimab targeting factor XIIa, navenibart targeting plasma kallikrein. We have additional oral therapies, sebetralstat as an on-demand kallikrein inhibitor, deucrictibant as a on-demand or prophylactic bradykinin B2 receptor antagonist. We have RNA-targeted therapies like donidalorsen targeted at plasma prekallikrein, and then gene therapies, which I'm not going to personally discuss, but you'll hear more about that therapeutic approach from my colleagues shortly. So to very quickly look at these investigational drugs that are newly developed and where we may be headed with HAE treatment of the landscape, this is garadacimab, the phase 3 study of this monoclonal antibody targeting factor XIIa. You can see in this placebo-controlled trial that there was about an 86.5% reduction in overall mean monthly attack rate compared to placebo with this once monthly subcutaneous medication. And on the right side, this important endpoint that we're increasingly considering, that 61% of the patients on the investigational drug were attack-free during the study period compared to 0 in the placebo group. This is phase 3 study data for donidalorsen, an antisense oligonucleotide that targets prekallikrein. And you can see on the left this phase 3 study data, 2 dosing regimens, one given every 8 weeks subcutaneously or given every 4 weeks subcutaneously. And at the bottom, you see that the mean attack difference versus placebo was at 87% from week 5 to 25, where this drug really starts to have its optimal effects. That was for the every 4-week dosing. You can also see towards the end of this study that every 8-week and the every 4-week start to approach each other. So it may be that every 8-week dosing gets you very much the same preventative effects. It just takes a bit longer to get there. On the right, this responder analysis, again, showing that 53% of the patients in the every 4-week dosing group were attack-free during this clinical trial. This is phase 2 study data for deucrictibant, and this is looking at long-term prophylactic therapy with the oral deucrictibant compared to placebo. You can see in this smaller study, because it's a phase 2 study, that there's about an 84.5% reduction in the mean monthly attack rate compared to placebo at this higher dose of oral deucrictibant at 40 milligrams a day. If you look on the right, these are attacks that required on-demand treatment, and you can see a 92% reduction compared to placebo with this oral approach to preventing HAE attacks. And then navenibart is a modified or engineered monoclonal antibody against plasma kallikrein. This has a long half-life, and based on the study data to date, may be able to be dosed every 3 months or every 6 months subcutaneously and still have good efficacy. This is, again, a phase 1b/2 study. You can see 3 different dosing cycles or cohorts studied, but again, in the 6 months that this was looked at, these preventative effects, greater than a 90% reduction in all of these cohorts compared to placebo, overall a 92% reduction in mean monthly attack rate. Now, we also have some advances in on-demand treatment, and we're focusing on long-term prophylaxis today, but I think it's important to recognize on-demand therapy may change in the near future as well. This is an oral plasma kallikrein inhibitor, sebetralstat, that was studied in a fairly large phase 3 study. You can see in this data just published in 2024, these significant reductions in the time to beginning of symptom relief compared to placebo at the 2 doses of sebetralstat, either a 1.6-hour time to beginning of symptom relief, 1.79 hours for the higher dose, compared to over 6 hours for placebo. And you see, again, similarly significant improvements in the time to the reduction in the severity of the attack for sebetralstat compared to placebo. An additional on-demand treatment is deucrictibant, this time studied as a rapid release or rapid onset formulation. This phase 2 study showing that this oral B2 receptor antagonist also seems to work very effectively to treat attacks. You can see these significant reductions in the VAS scores at 4 hours after treatment compared to placebo. Statistically significant for all the 3 doses studied here, and a dramatic reduction in the need for rescue medication down to 6.5% of patients at the highest dose compared to over 60% of patients that needed rescue to treat their attack in placebo. And so we have these advances coming along. What I haven't covered here is, of course, gene therapy, which you're going to hear about in a moment. There's also earlier phase developments for small interfering RNA and some other oral agents that may be useful for preventing attacks in the future. Now, of course, what I've shown you are new drugs or investigational drugs, and we need more data on safety, efficacy, tolerability, and of course, quality of life. We have some very strong data from the phase 3 studies that I've shown you, but of course, more long-term data will be important as we move ahead with these additional therapies. So I'll finish just by saying that I think these advances in therapies are moving us closer to some of the goals in managing HAE. One of the many important contributions that our dear friend Marcus Maurer had in this field was this Delphi initiative that demonstrated that experts agree we should be aiming to normalize patients' lives, we should achieve total control of the disease, and we should seek input from patients on whether they consider their HAE to be well controlled or normalized, which again speaks to patient preference and the importance of burden of treatment and access to treatment. And so I think any or all of these therapies will help move us closer to this total control of disease. With that, I'll thank you for your attention, and I'll turn it back over to the program moderators. Thank you.
So, Danny, you're up next. Thank you.
Thank you very much. Good morning, good afternoon, good evening, or even good night. It's a pleasure. My name is Danny Cohn, and I'm very pleased to present CRISPR technology. What is it and how does it work? These are my disclosures. And as Marc already highlighted, indeed, the current guidelines recommend as an ultimate treatment goal normalization of patients' life. But there can be many definitions how we normalize patients' lives. Naturally, patients want to get rid of their angioedema attacks. They need adequate acute treatment. They wish to have no need for short-term prophylactic therapy. They want to be independent of caregivers. They want to do whatever they like, so no restrictions of activities, but maybe ultimately, as the tip of this pyramid, no need of prophylactic treatment reminders, so a one-time treatment and maybe even a functional cure of the disease. and that is where gene therapy comes into place. What I would like to discuss today is first to define what is gene therapy and also introduce CRISPR therapy. We will particularly focus on the applications of CRISPR in genetic diseases, particularly hereditary angioedema. I will explain the mode of action of NTLA-2002, which is the first CRISPR-Cas gene therapy under investigation for hereditary angioedema. We will definitely also emphasize on how we can mitigate the potential risks of the NTLA-2002. And then I will conclude with the trial design of the clinical development of this treatment. And I look forward to the discussion and answering your questions. So gene therapy can be defined by various mechanisms. First of all, a viral vector-mediated gene addition, where a viral vector containing a small part of DNA, which is added to the human genome as an episomal DNA, for instance, in attempting to increase the C1 inhibitor levels in patients. We know that this way of gene addition has been successful in, for instance, treating patients with hemophilia B, where only a very small amount of increased amount of target protein is needed to have a reduction in severity of the condition. For hereditary angioedema, this has been attempted in a phase 1 trial, but unfortunately there was no sustained increase in C1 inhibitor expression. And please be reminded that for getting rid of angioedema attacks, a high increase of C1 inhibitor expression is required, maybe more than 40%. Another way how gene therapy can be applied is by very specific gene edits at a target gene. This is, for instance, with CRISPR-based gene editing. And we define both in vivo and ex vivo applications of CRISPR therapy. Let's focus a little more on that. So in vivo gene editing is where CRISPR is the therapy. CRISPR is administered in a one-time IV treatment to the patient, and the gene editing occurs within the body of the patient following this administration. That is distinct from ex vivo gene therapy, gene editing, where CRISPR creates the therapy. Stem cells are harvested from the patient, are treated with CRISPR, and then following conditioning with chemotherapy in the patient, re-administration of these stem cells. Gene editing research is not completely novel. The very first description of CRISPR stems from 1987, where it was discovered in E. coli. And in 2013, there was the first report of gene editing in human cells by Jennifer Doudna and Emile Charpentier, who were awarded with the Nobel Prize for Chemistry in 2020. Already in 2021, there was the first in vivo CRISPR therapy phase 1 results, and in 2023, FDA approved the first ex vivo application of CRISPR therapy for patients with sickle cell disease and severe beta thalassemia. And last year, the first in vivo CRISPR phase 3 trials were initiated and are still ongoing. There are several purposes for which CRISPR-based gene editing can be used. For a gene knockout, where a disease-causing protein needs to be reduced, this can be achieved with a lipid nanoparticle that carries the CRISPR therapy. But also repair or insertion of functional DNA can be used. In this case, since this requires a larger vehicle, both a lipid nanoparticle and an adeno-associated virus are required. As Marc already highlighted, we know exactly which components contribute to the occurrence of an angioedema attack in hereditary angioedema, and we also know that various components are currently the target of new treatments. C1 inhibitor is, of course, the disease-causing protein in this case, and would be a logic target for CRISPR-based therapy. However, there are many downsides. First of all, the SERPING1 gene is relatively large and does not fit in the small packaging capacity of AAVs. Secondly, there are many potential disease variants. So a one-size-fits-all CRISPR therapy targeting the C1 inhibitor gene is very challenging. And lastly, investigational gene editing therapies introduce a small precise edit in a gene that is disease associated. So that makes also SERPING1 a difficult target. To the contrary, we know that kallikrein is a clinically validated therapeutic target for hereditary angioedema, both for prophylaxis but also for acute treatment. And we know that inhibition or decreasing plasma kallikrein decreases the rate of angioedema attacks. And therefore, this protein is very attractive also for CRISPR-based gene editing. Let's have a closer look at NTLA-2002. On the left, you can see an enlarged image of the lipid nanoparticle that is coated with apolipoprotein E. Apolipoprotein E has a very high affinity to the LDL receptor, which is expressed on liver cells. The lipid nanoparticle contains two components. First, the single guide RNA, which very specifically guides the CRISPR therapy to the targeted place at the genome. Furthermore, it contains messenger RNA encoding for Cas9, which is the machinery that performs the gene edit. Following a one-time IV administration, the lipid nanoparticle binds to the LDL receptor, and following endocytosis, the messenger RNA is translated to Cas9, and this forms a complex with the single guide RNA. The single guide RNA Cas9 complex, or also known as the CRISPR-Cas9 complex, subsequently enters the nucleus. And we will now see on the next slide what happens when this complex enters the nucleus. The single guide RNA very specifically localizes the target place at the DNA where the gene edit is needed. And Cas9 performs this gene edit. And following a double-stranded break in the DNA, natural repair mechanisms by the body repair this edit, this cut, by non-homologous end joining. But this is error-prone repair, and this leads to small disruptions in the KLKB1 gene. So KLKB1 is the gene encoding for plasma prekallikrein. And this occurs by small insertions or deletions, which are called indels, and this finally results in reduction of functional prekallikrein messenger RNA and subsequently also a decrease in plasma kallikrein levels. How do we know that this treatment is safe? So I would like to emphasize now the preclinical work that has been done before the NTLA-2002 was tested in human beings. First of all, we needed to very carefully select the right single guide RNA to avoid any off-target effects. One of the ways how this was assessed was by very in-depth in silico analysis, where the identification of potential off-target effects was assessed and the optimal single guide RNA was selected. Secondly, in animal models, naturally toxicity analysis had been performed, but also it was assessed whether only the somatic gene edit was performed and the germline cells were not affected, by also testing the offspring of these animal models. And further, in vitro analysis were performed on human hepatocytes that were treated with doses which were up to 50 times as high as the intended dose in human beings or in humans, to also test these cells for any off-target effects. And in short, this risk, or based on these experiments, seems negligible. Furthermore, I would like to highlight that NTLA-2002, due to this ApoE coating, has a very high tropism to the liver, which further reduces the risk of off-target effects in other organs. And lastly, we know that prekallikrein reduction or kallikrein reduction is not associated with morbidity in humans. There is a certain condition which is called Fletcher's trait, where people lack the complete prekallikrein gene, have a homozygous mutation where they lack kallikrein in the plasma, and they have a normal life expectancy without any other morbidity. So reduction in prekallikrein or kallikrein is not associated with morbidity. Let us now focus on the trial design for NTLA-2002. So I would like to remind you once more that this is a one-time treatment aimed at a functional cure of hereditary angioedema by knocking down the prekallikrein or kallikrein levels. Patients, first of all, there was a phase 1 study, and I would like to highlight that this was also done in patients with hereditary angioedema because it's not ethical to treat healthy volunteers with a gene editing therapy. This was an open-label, single ascending dose study where patients with hereditary angioedema were treated with either 25, 50 milligrams, or 75 milligrams of NTLA-2002, and the primary endpoint was safety and tolerability. The key secondary endpoints included kallikrein levels and naturally also clinical efficacy assessed as angioedema attack rates. In the phase 2 study, this was a blinded placebo-controlled study where patients were treated in a 2:2:1 ratio with either 25 or 50 milligrams of NTLA-2002 or placebo. And the primary endpoint was clinical efficacy, defined as the number of angioedema attacks per month during the primary observation period, which lasted between weeks 1 to 16. And the key secondary endpoints included again safety, the number of angioedema attacks per month when the new steady state was reached and the pre-existing prekallikrein or kallikrein was consumed, and the change from baseline in total plasma kallikrein levels. And as Connie already mentioned, Hilary is going to present these data on these study results after my presentation. But first of all, I would like to thank you very much for your attention, and I'm happy to take any questions.
Thank you very much, Danny, for a very understandable presentation on difficult principles. While we're waiting for our participants to write in with their questions, I think it's important to say, of course, you mentioned this is a functional cure and patients still carry the genetic risk. So children, offspring of those with HAE still have the capacity to develop HAE. That's correct, isn't it?
Yes, that is correct. Indeed, the targeted gene edit only occurs in the liver. We know that the treatment is sustained. At least Hilary is going to present the latest follow-up data, but we know that the daughter cells of the treated cells, and I mean with daughter cells, hepatocytes from that keep dividing during patients' lives, that also the divided cells contain the gene edit. However, the germline is not affected, and that is known from the animal model work that indeed this is only a somatic mutation in the hepatocytes that does not infect the germline. So children from patients with hereditary angioedema that receive this treatment are still at 50% risk to also inherit the disease.
Yes, thank you. Now there's a question about liver disease. Firstly, if a patient with HAE has significant liver disease, is this still a treatment that they could consider? And secondly, what happens to a patient who's had this treatment as life goes on, develops severe liver disease?
So this is a very good question. Naturally, we're still at the early development of this treatment, and in phase 1 and phase 2, patients with severe liver disease were not eligible to participate. Fortunately, liver comorbidity does not occur very frequently in patients with hereditary angioedema nowadays, although we know that in the past the risk of viral hepatitis was increased when patients received the older plasma-derived products. We need to learn in the future if patients with, for instance, a viral hepatitis are also eligible for this treatment. We did not observe any hepatotoxicity in the patients that were treated so far. So we need to learn in the future for a more definite answer, but I don't think that there is a very true contraindication for patients with mild, for instance, NAFLD fatty liver disease to also receive this treatment. The second question was about if there is if patients develop liver disease. That is something that we need to learn from long-term follow-up studies, but I don't expect that there's a particular risk of loss of efficacy if patients are will develop a severe liver disease in the future.
Thank you, Danny. We have another question from Professor Hide, and it's, is there any possibility that populations of edited cells in the liver may change over time, lifelong?
So we have observations now for a very prolonged period in both animal models, but now the first year is also in humans. And so far, we haven't observed any loss of efficacy because, as I tried to explain earlier, the daughter cells of the hepatocytes also carry the gene edit, and also these daughter cells are unable to produce the normal plasma prekallikrein. So we assume so far that plasma prekallikrein is sustainedly reduced in patients that have this gene editing therapy.
Well, thank you very much, Danny, for your presentation and for the question and answer session. We'll now move on to Hilary's presentation. I'm sure we're all looking forward to hearing the latest update on the trial patients in this very exciting studies. Thanks, Hilary.
Thank you very much, Connie, and thank you for inviting me to speak. So I'm Hilary Longhurst from Auckland, and I'm going to talk about the first 37 patients who've been treated so far. And I'm happy to say that Connie, Danny, and I were among the first investigators to be able to use this therapy. So you've seen this before, but just to say that I'm going to talk about both the phase 1 and the phase 2 study together, just to save time. The phase 1 was open label. People were able to continue their long-term prophylaxis. But after 16 weeks in this study, people could choose and did choose to stop their prophylaxis. Phase 2, people had to be off long-term prophylaxis in order to take part. So these are the people who took part. Just going to move myself out of the way slightly. And you can see that both genders, males are slightly overrepresented. This is actually the double-blind phase, but generally fairly balanced. There was a large weight range from pretty petite, 50 kilograms, to gentle giant, 133 kilograms. And this is not a weight-based treatment, it's a single-dose treatment, both HAE types. Most people had used long-term prophylaxis previously, and there was a variety of different prophylaxis used prior to the study entry. Although, as I said, everyone had to come off their long-term prophylaxis with a 5 half-lives washout before starting on the study. And the number of angioedema attacks had to be at least 1 per month. And for the participants, the number in the historical attack period was from just over 1 a month to just under 2 a week. And as one would expect, the typical attack severity was moderate or severe in most cases. So I'm going to start with showing you the reduction in plasma kallikrein, because this really demonstrates to me what we were seeing. In New Zealand, we had quite a few patients who were on this middle dose here and quite a few who were on placebo. But you can see this is kallikrein levels over time with the first 16 weeks here, but we've got up to almost a year in this data cut. And you can see that those on placebo did not have any significant reduction in their kallikrein protein levels. Those on the top dose, shown in this bottom blue line here, had a very profound reduction in plasma kallikrein, and it's way below. You can see the dotted line here at 60% reduction, which is the level at which we think, below which we think people will not have, or very, very unlikely to have HAE attacks. So that's really our target. And then the 25 milligram dose, you can see that many people did eventually get below the 60%, but not everyone. And they took a long time to settle down. And this is what we felt we were seeing actually in practice before everything was unblinded. We felt that there was a clear dose effect, but people did tend to improve over a longer time than perhaps one might expect. So this again is the phase 2 data, and you can see here that the data is very good, up there with the other modern prophylactic treatments. And you can see for the people treated with both doses, there's a large reduction in mean attacks per month, with a bit of variability, but clearly less than placebo. After the first 4 weeks, the reduction is even better, 80 or 81%, depending on the dose, compared with placebo. But this is all very good. But remember, Marc has told us that actually our aim is absence of attacks and normalization of life. And although 8 out of 11 patients in this group, in the high dose group, were completely attack free, you can see here, and 4 out of 10 in the low dose group, there are people who are still getting attacks, and I'll come to that in more detail in a minute. You can also see that by other methods of attack reduction, there does seem to be this dose dependent effect, whereas the low dose isn't quite as good as the high dose compared with placebo at reducing the frequency of attacks. And this, I guess, is not too surprising in a placebo-controlled trial, but in the phase 1 trial where it was open label, everyone knew what they'd had, pretty much everyone was attack free, and even in the first 16 weeks we had reduction in attack rates of 82 to 95%, with the total on study period being close to 100%. So before I go on to the detail, I just want to look at the quality of life, which of course is very important. So you can see here in the grey, this is placebo, and although there have been some reductions in angioedema related quality of life for the placebo participants, so lower is better, improvement, they've not reached the clinically significant level of a reduction in 6 points, except fatigue and mood might have just squeezed in here. But you can see for both doses, there's really very clinically significant reductions, again with this dose-dependent effect in favour of the higher dose, as one might expect. And then when we look at work productivity, clearly we want people living normal lives. This means that they're back at work, they're contributing to society, and they're not impaired with their normal non-work social and family activities. And you can see with placebo, things actually seem to have got worse. There's more absenteeism, there's more presenteeism, which represents the time that our participants are spending at work, but not feeling 100% and maybe not 100% contributing compared to where they would be if they had no angioedema. There's a significant productivity loss and activity impairment even out of work for the placebo group. And that seems to have worsened over week 16. And I can tell you from the New Zealand experience of having 3 people on placebo, it was a very, very difficult time for both investigator and, more importantly, for the participants who'd clearly were hoping so much to have a really effective treatment. Conversely, those people who were treated with active therapies, they had a significant reduction in work impairment and improvement in productivity, presenteeism, and improvement in reduction of activity impairment. And again, this looked to be dose dependent, with the higher dose being better. So I guess when you infuse intravenously a lipid nanoparticle with some RNA in it, there is a big worry that it might not be very well tolerated. We did give a pre-med the night before and on the morning of the study of dexamethasone to prevent infusion-related reactions. And we, in all but one case, gave intravenous C1 inhibitor because we predicted that a lipid nanoparticle is just the sort of thing that might activate complement and trigger an HAE attack. In fact, I understand that the patient who didn't have C1 inhibitor also was fine. This is the phase 2 data, but what you'll see here is that there were some infusion reactions. These were pretty mild. The worst were fever. 2 of my patients had Vigors, which were terminated with extra doses of corticosteroid. Someone had severe back pain, which meant they had to temporarily stop the infusion. But these were all easily manageable and the sort of thing that one might encounter in an afternoon in outpatients when giving such an infusion, and certainly didn't need any more treatment other than slowing or stopping of the treatment and extra steroid and symptomatic treatments. In the phase 1, 7 out of 10 people had infusion-related reactions, but again, nothing too severe. And I like to think that we got a bit better at learning how to give this treatment to minimise that reaction. And then other than infusion reactions, the adverse events are really what you'd see for people living in New Zealand, Europe, sort of in 2021 to 2024 when this study was done. So really very reassuring. When you give the infusion, you do see a little blip in CRP and a little blip in liver function, but for the vast majority of people within the normal range. So again, no clinically significant laboratory abnormalities. One of my patients on 3 weeks after having had the treatment went to a Maori wedding, and although our patients are advised to have a limited alcohol intake, 1 drink per day maximum for the month after the treatment, it's possible that at a Maori wedding more alcohol might be served. And this patient did have a little blimp in ALT, which returned to normal relatively promptly without any treatment. He was asymptomatic throughout. So just to drill down into more detail in how the patients have been. So I love this graph. This is the first 10 patients, so open label, and each horizontal grey bar represents a patient. You can see that there were 3 doses here. You can see their monthly attack rate prior to screening. And on these bars, there are these tiny vertical bars. Each one of those is an attack, colour-coded for severity, and the width of the bar represents the duration of the attack. And you can see that with one exception, people had quite a few attacks prior to treatment, which is this horizontal solid bar at time 0. But within the 16-week observation period, pretty much everyone had stopped having attacks. There's 2 provoked attacks here. This one wasn't treated, this one was treated, but nothing since. And we do now have another year's results, which will be published. I think they're going to be presented at the EAACI meeting, so I can't wait to tell you about that. And the other thing to say is that everyone who had been on prophylaxis, so in this case it was attenuated androgens or berotralstat, they were able to withdraw their prophylaxis without return of attacks. So we were extremely encouraged at this. Now looking at the phase 2, as I said, there was an encouraging reduction. It was up there with the current first-line treatments, but it wasn't complete absence of attacks. So let's look in more detail. And I think the first thing you can see on this is that you can see the dose-dependent effect. At the bottom are the people having placebo, and you can see here this is very busy with attacks. It's less busy after the 16-week observation period because patients were allowed to restart prophylaxis, and these black diamonds are when each patient restarted prophylaxis, which didn't work brilliantly, as you can see. On the right-hand panel, you can see the reduction in kallikrein levels. And if I look at this, this top patient here has got a very good reduction in kallikrein levels, but she's apparently got a 29% increase from baseline in attack rate. She's my patient, so we went back to her and we said, well, you've been on active treatment, but you're still having attacks. You know, what's going on? And she said to us, well, what are you talking about? My life's transformed. I'm looking after my children. I've taken on a part-time job and I'm training to be a lawyer. You know, my life's completely changed and the attacks are very easily treatable. And in fact, what had happened with this lady was that her attacks were mainly laryngeal. She was a smoker, so whenever she got a sore throat, which was frequently, typically in the past she would have had a laryngeal attack. So she used to treat these attacks very, very promptly. Once she knew she'd had the active treatment, she took this slightly scary step of waiting to see what happened, not normally advised. And in fact, it turned out that her symptoms were a normal sore throat, which did not progress to an angioedema attack. And she's since had several other triggers for attacks which have not progressed. And in fact, since we had the conversation with her, which was at the end of this arrow here, she's had no further attacks and she's stopped prophylaxis again. Patient number 2 is actually Danny's patient, so I may let him speak to this, but this was a bit of a different situation. This was someone who did appear to have visible peripheral attacks, but has subsequently been attack-free while going back onto lanadelumab, which I think was her pre-treatment prophylaxis. So who knows what the situation is here. And a similar sort of situation with this patient here, who is a French patient. These patients have all been retreated, and I think I have to wait till further time to tell you the happy news about how they are now doing. So that's just comparing the full dose treatment, which we've now selected as the optimum treatment, with the placebo, and you can see there's a clear difference with the majority of people being attack free. So in conclusion, we think that the inactivation of the prekallikrein KLKB1 gene in the liver is akin to genetic surgery. It's very specific for the liver and for the gene. We believe it's safe, though there is a long-term follow-up, and we believe it's permanent. And we're definitely not seeing any concerning reductions in kallikrein or any return in the kallikrein level that would give us concern that was the case. So we think it's permanent. We find that it's very effective in treating symptoms. We think that many, if not all, eventually of these patients will have a functional cure. So far, it's well tolerated. There's no prolonged immune suppression, just the pre-med, and with corticosteroid and of course C1 inhibitor. The treatment is it's very simple science, it's very elegant science, and it's potentially very easy to manufacture. So HA is leading the way again, but of course this is not the only disorder where this treatment could be effective. We know, as Danny said, that the treatment could be modified to treat other genetic diseases where a gene needs to be knocked out or inactivated. And this rare type of amyloidosis is also under trial and looking very promising indeed. This treatment is in trial for hereditary hyperlipidemias, but could also be used for more common hyperlipidemias. So a single treatment instead of years on statins or other medications. We know that the treatment already has been used ex vivo, doing blood cells, but potentially with different delivery methods or adeno-associated virus directed delivery. We could use it to treat other parts of the body. And as Danny said, similar treatments can be used to insert, correct, or even reversibly silence genes. And examples now going into trials on hepatitis B and alpha-1 antitrypsin deficiency, where these approaches are being tried. So this is really very exciting technology. And with my immunologist hat on, I'm looking forward to development to deal with a whole load of the other rare diseases that we deal with, not to mention some common diseases. So just to thank everyone who's taken part in the studies, New Zealand Clinical Research and my colleagues in Auckland who've been looking after the patients, Intelia who've developed the technology, and of course, more than anything, our study participants and their families who've stepped forward to be the first people in the world to receive these therapies. So thank you very much, and I'll be delighted to take questions.
Thank you very much, Hilary. Since you invited Danny to comment on his patient that showed reactions on the infusion, Danny, can I ask you to explain what you think was going on?
Yes, so this patient experienced severe angioedema attacks prior to her participation, also when using long-term prophylactic therapy, which was, by the way, C1 inhibitor plasma-derived concentrate. And we will see in the new update, in the new data cut, how this patient is doing. This is data that has not been published yet, so therefore I cannot share the entire follow-up. But indeed, since this patient still experienced mild angioedema attacks during the follow-up, we installed her with a plasma kallikrein inhibitor. This is to be followed up soon, hopefully.
Thank you very much. Well, I'm afraid we've come to the end of our seminar, and I'd like to just remind all participants that we'd like you to complete the feedback form at the end of the webinar in order for you to receive your CME points, and there is a QR code there for you to scan. Our next webinar will be held on May the 7th, and it will examine the dynamics of triggers and prodromes in hereditary angioedema, another fascinating topic for HAE specialists. Stay connected with on socials and the website. And it remains for me to thank Hilary and Danny and, of course, Marc for their contributions to what I think has been a very exciting seminar. Thank you all for your interest and participation. Good morning, good night, good evening.